Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Amira Farzana binti Kiri Ismail and my number matrix is 208194. Next, Haniza.
Okay. Assalamualaikum and hi everyone. My name is Nur Haniza binti Marsid and my matrix number is 205477. Okay, next is Noraini.
Hi, my name is Noraini binti Mustafa. My matrix number is 206669. Next is Nuraini.
Hi, my name is Nuraini binti Mokhtar with matrix number 205304. Next, Fagira.
Hi, my name is Fagira binti Ahmad Ahmad and my matrix number is 205275.
Alright, thank you everyone. So today we will be discussing about surface sterilization of Splen. So may I start first? Alright.
Yes.
This is our data recording. So the first question, based on the observation, explain the result obtained. Suggest a modification, if any, to improve the protocol. So I will explain the table. So as you can see here, our sterilizing agent is 10% chloride solution. So our group must do 10% chloride solution and the exposure time given is 15 and 5 minutes. So I assume that the number of Splen we prepare is 5 for each exposure time. So as you can see in observation, the number of contaminated Splen in percent is for 15-minute exposure time is 0%, while the number of viable Splen remain green is 80%, and the number of dead Splen turns brown is 20%. So I assume that this plate is not contaminated. While in the 5-minute exposure time, the number of contaminated Splen is 60%. The number of viable Splen remains green is 40%. And lastly, the number of dead Splen turns brown is 20%. So in this plate, I assume it is contaminated. So how do I get the number of percentage such as 0%, 80%, and 20%? So as you can see here, to calculate the percentage is for percentage contamination, the number of contaminated Splen divide with the number of Splen prepared and then times with 100%. So for example, in the 5-minute exposure time, the number of contaminated Splen is, how do I get 60? 3, I assume there is 3 number of contaminated Splen. Divide with 5 and times with 100%. And then I get 60%. So in this, I got 4 viable Splen, and this one I got 1 dead Splen. Same as the others. So next is suggest a modification, if any, to improve the protocol. So there is only one suggestion, which is, in my opinion, during the cleansing or the rising of the leaves, I will include soap. Okay, we will wash the leaf with soap first so that we can remove the unwanted chemical agent or wax on the leaf. I'm afraid they may interfere with the sterilizing agent. So that's why this is my suggestion for the modification to improve the protocol. So for the second question, based on the result, draw a suitable conclusion. So the efficiency of sterilization is affected by the amount of exposure time of Splen with sterilizing agent, which is for our group is 10% chloride solution. And I assume we can achieve 100% viable Splen, but if the amount of exposure time is suitable. However, an excessive amount of exposure time of Splen with 10% chloride solution may result in dead Splen as they cannot handle the amount of chemical solution. That's why if you can see in the 15 minutes exposure time, there is one number of that explant. Because maybe the explant cannot handle the chemical agent in the Clorox. So it will result to that explant. So I think that's all from me. Is there any question?
May I add some points for modification question number one?
Sure.
Okay, we can also stir the explant while it is being disinfected to enhance the contact of the explant with the disinfecting agent. And second is we also can frequently add surfactant, that is twin 20, to the sodium hypochlorite. Also the same to increase the contact between the explant and the disinfectant agent. Okay, that's all.
Thank you, Haniza. I agree with you too. So is there anyone would like to continue number 3?
Can I continue?
Sure, Aini.
Okay, so I'm going to answer question number 3, 4, and 5. If it is okay?
Yes.
Okay. Question number 3, apart from exposure time, explain another factors that may affect the efficiency of the sterilizing agent that we use in this practical. So my answer, I provide 3 answers for the 3 factors. The first one is the type of the leaf surface, because some leaf surface may contain wax that prevent the sterilizing agent to penetrate through the leaf. And the second factor is the physiochemical environment, where the increase in pH will improve the antimicrobial activity of the disinfectants while reducing the antimicrobial activities of other. And the pH affects the efficiency by altering the disinfectant molecule of the cell surface. And the last factor is that temperature. Some disinfectant can increase its activity when exposed to high temperature, but there's also disinfectant that's too great and increase in temperature that causes it to degrade. Okay, moving on to question number 4. If surfactant is not used, predict the outcome of the practical based on the percentage obtained above. So we need to know what is the purpose of using the surfactant. The purpose is to reduce the surface tension and allow the chemical agent to react with the whole surface of the plant. So I think the percentage will decrease and the surface tension cannot be reduced. So the Clorox it cannot penetrate the fatty substance. Okay. Question number 5. If the step that treated the explant with 70% of alcohol was ignored, could the outcome of the practical be changed significantly? Explain. Okay. If it is ignore the number of if we ignore the 70% of alcohol, the number of contaminated and that explant may increase. 70% of the alcohol is used to help us to kill the microbes, the bacteria, and other microorganisms on the surface of the explant before we transfer it into an in vitro environment. So since there is no ethanol, the probability for the culture to contaminate during the in vitro transfer is high. I think that's all from me. Does anybody want to continue to question number 6?
Yes, I would like to continue question number 6, 7, and 8. So for question 6, suggest a suitable sterilizing agent to replace Clorox used in this practical and modify the protocol to suit the new sterilizing agent. So for me, the suitable sterilizing agent that can replace Clorox is calcium hypochlorite. It is commercially available in the form of powder. So how we can modify the protocol to suit the new sterilizing agent? When it is in the form of powder, before we use it for sterilization, it must be dissolved in water. So after it dissolving in the water, we filter the solution. So the final concentration of the solution is in lab around 3.0%, and the concentration is just same as the sodium hypochlorite. Then for question 7, name the type of sterilizing agent used in this practical. The type is, it is a laundry bleach. Actually, it has many type of sterilizing agent. The other type is in the type of chemicals such as mercury chloride and hydrogen peroxide. But both of these chemicals, it is rarely used because it's too dangerous, such as hydrogen peroxide. The concentration is too high, which is 30%. Then number 8, name the active ingredient of Okay, I will proceed to question number 8. Name the active Clorox and its concentration at 10% solution of Clorox. So Clorox is known as commercial bleach. So commercial bleach usually has 5% concentration of sodium hypochlorite. But for practical culture, Clorox here will be diluted to 10-20%, which result in 0.5 to 1.0% of the at minimum concentration. So, in this experiment, the exposure time that we use is so 0 to 20 minutes. It's enough to do surface sterilization. So for more information about the concentration at 10% at question number 9. Thank you.
Okay. Okay, may I proceed for question number 9 until 11? Explain the procedure to prepare 10% volume of solute over volume of solution of Clorox solution. Mention whether you need to autoclave the solution or not. Justify. 10% Clorox solution is prepared by adding 10 ml of Clorox solution to a graduated cylinder and then diluting with water to 100 ml with a few drops of Twin 20. The duration of treatment is usually from 5 to 30 minutes, followed by 3 to 5 rinses with sterile water. Okay, why we uh, why we do not autoclave the Clorox solution? Because Clorox solution can produce toxic product that corrosive the autoclave and also degrade the hypochlorous acid. Okay, question number 10. Justify the need to rinse and expose the explant with sterile water for a certain period of time after Clorox treatment. So this is because we want to remove the sterilizing solution and also prevent explant from contamination. For question number 11, if rinsing with sterile water was performed quickly without leaving the explant in water for a while, what would be the consequence? It will cause the retention of the sterilizing solution that will become a toxic chemicals towards the explant and also damage the explant. Therefore, the percentage number of viable explant remains green is decreasing, while the percentage number of contaminated explant and the percentage number of dead explant turns brown are increasing. And then the damaged tissue has to be removed before culturing the explant. Okay, that's all for me. Anyone want to answer question number 12?
Okay, I will proceed with number 12. Okay. For number 12, if the source of explant was not heavily contaminated, which part of sterilizing protocol could be modified to make it more effective? Explain. So I think for the part of sterilizing protocol that could be modified is the surface sterilization with ethanol step. For the surface sterilizing, sterilization with ethanol step can be changed to T2 treatment. The best result were obtained using the highest concentration of sodium hypochlorite in T2 treatment. It is well known that the sodium hypochlorite have a broad spectrum of antimicrobial activity, and it is able to rapidly kill vegetative spore, bacteria, fungi, protozoa, and viruses. Sodium hypochlorite exerts its antibacterial effect by inducing the irreversible oxidation of sulfhydryl group of essential enzyme, and it may have deleterious effect on DNA and membrane-associated enzyme activity. For the number 13, if the source of the explant was sterile, which part of the protocol could be modified? Explain. For sterile explant, plant tissue culture can be made directly by performing aseptic techniques properly for preparation of explant, and explant culture without cleaning plant part for explant preparation and performing surface sterilization procedure. If the explant if the sterilization process of explant have been doing again, it will be damaged and not show potential to regenerate during the shoot and root inducing step because of excessive use of sterilizing agents such as Clorox and of Clorox solution and mercury chloride. For the number 14, suggest a method to grow a plant under sterile condition for explant preparation. I think the method to grow a plant under sterile condition for explant preparation is using surface sterilization. For the surface sterilization is the process of which involve the immersion of explant into appropriate concentration of sterilizing agent for a specific time that will resulting in the establishment of the contamination-free culture. If the sterilization fail, it will have a little effect in cell division and it will restrict growth and development of the explant. I think that's all for my answer. Anyone want to add something?
I think there is no for me.
I want to ask something. For question number 6, why do we need to add water in the sterilizing agent? Can you explain?
It is because potassium hypochlorite are available in the form of powder. So before we use it, we must dissolve it in water.
Okay, thank you.
Can I add for question number 14?
Yes.
This is from my research. We can also do explant pre-treatment to control microbial contaminants such as heat treatments. Heat treatments can be effective for some viruses as well as fungi and bacteria. For example, potted plants can be put in a growth chamber with the temperature at 35 to 40 degrees Celsius for prescribed time periods in order to rid some plants like potatoes of viruses. And second, a fungicide treatment of plant material prior to culture may help reduce fungal contamination. Before use these heat treatments, it requires studies to determine the method of heat most adaptable to the plant material to be treated and the combination of time and temperature. Therefore, to ensure that the host is not harmed but effective enough to kill the conserved pathogen in the plant part by using these heat treatments. Okay, that's all for me.
So is there anyone that would like to add any more?
No, for me.
Okay, thank you, Aini. Fagira? All right, Raini Mota
No.
and Haniza.
No.
All right. So I think that's all from